W. Ball-Tibbs special report on a recent study that discusses a new scientific breakthrough.
That's right, Cal, and that scientific breakthrough is the process called gene therapy. Some researchers are saying that it may be the first step to curing cancer.
Let's roll our footage from a recent interview with one of the researchers, Dr. Crum. How do you pronounce the name? It's Crumwiak. Crumwick? Whatever, Mr. Producer. Play the tape.
I've heard that your research relates to something called melanoma. Could you explain what this is?
Sure thing. Melanomas are skin tumors, and they result from mutations in the skin cells. A mutation is basically any unusual change in the DNA code sequence. Cells mutate all the time in just about everyone on Earth, but the mutations usually get rooted out and stop before they can spread and cause harm. Melanomas occur when that process doesn't happen and the mutations run rampant.
Could you explain what sort of factors will allow the mutation to spread?
Whenever a cell's DNA is exposed to something called carcinogen, the risk of cancer tends to increase. There's a specific gene called the tumor suppressor gene that keeps the cell from dividing too much, especially if the cell is mutated or incorrectly coded. Carcinogens target this gene, preventing it from carrying out its proper functions.
What are some specific carcinogens?
Some of the most common carcinogens are asbestos and cigarette smoke. There are over 100 known carcinogens in the environment. One of the biggest ones is ultraviolet radiation, which comes from sunlight. About 85% of the melanomas that people get are caused by too much exposure to sunlight.
Are you saying that people can get cancer from the sun?
Well, a little bit of exposure won't hurt anyone, but the risk for melanoma doubles when a person has had more than 5 sunburns. Plus, it's less common among people with higher levels of melanin in their skin. That means people with darker skin are going to be more protected against the cancer. And people with more pale complexion are more likely to be in danger.
Could you tell us about your recent study on gene therapy? Explain the process and what it means.
My lab partner, Dr. Proctor, actually did a lot of the experimentation, but I can tell you what we found. We isolated a gene which we call P53, and it is a tumor suppressor gene that gets mutated in 80% of melanoma cases. We figured this gene was a big factor in both the formation and also the prevention of melanoma. Using another gene that creates a glowing green protein every time the P53 gene was used, we were able to record how much of the cell was glowing and when it glowed during the cell's life cycle.
How did you implement this?
We took some lymphocytes, which are white blood cells that fight tumors, from melanoma patients. We inserted a gene that codes for anti-cancer protein called the tumor necrosis factor right into those lymphocytes. The altered cells were able to successfully kill off the mutated cancer cells of the melanoma patients.
Thank you, Dr. Cromyak, for your time. We know you're very busy. Coming up next time, we will interview Dr. Proctor, the assistant scientist, to hear how the two researchers plan to use their study in cancer treatment.
We're live in the studio with our special guest, Dr. Proctor.
Thank you for having me. It is a pleasure to be here.
We just finished filming an interview with your research assistant, Dr. Crum something.
We would like to know how you both plan to use his findings for actual medical treatment.
I don't know if Dr. Chromiak mentioned gene therapy, but that is essentially what we are looking to see as an implementation for our research. It would have to be left to the individual drug companies and pharmaceuticals to figure out the details. But we basically would like to see our modified form of the P53 gene available for commercial use. Cancer patients would be able to pay for a treatment that actually implements the gene directly into their own white blood cells. Doing so would make the white blood cells adapt the trait responsible for killing cancerous tumors and growths.
That is very interesting. Why not just let the cancer cells die on their own?
Well, one of the things that makes a cancer cell so dangerous and deadly is that normal cells with damaged DNA will die naturally, but cancer cells with DNA will not die unless they are actively attacked and killed. That's why our form of treatment is so important. We are basically sending in a little immune system army to fight the cancer and destroy it in the body. It's also better than some other treatment methods, like chemo, because it is very specific to the cells it attacks. Chemo and radiation therapy damages hair, muscle, and organs like the liver. Our method is a lot safer. It only attacks tumors, meaning that there are a lot less undesirable side effects.
It sounds like this kind of discovery could be very, very, very important.
I do hope that other researchers will be able to take what we've learned and start implementing it for public use.
Thank you again for coming to our news station for this interview.
No problem. Thanks again for having me. You said it so weird.
This first chart shows how the amount of something called tumor necrosis factor changes over time. Tumor necrosis factor is the gene that codes for an anti-cancer protein. As you can see, the tumor necrosis factor increases as time goes on. The second chart shows the percentage of cells in the skin tissue sample with the mutant p53 gene. The mutant p53 gene kept cells from dividing too quickly, and the percentage of cells in skin tissue decreases.